So because of this revolution, though, there's no question genomics will have impact on medicine. And it already is. And I'll start out by telling you a little bit about that. There are 3 areas in which sequencing your DNA will have enormous impact. One is in understanding and treating certain kinds of diseases. And I'll go through this in a little more detail in a minute. But genome sequencing is having enormous impact in the area of cancer. What I call mystery or undiagnosed diseases, that's another area where it's having some impact. It's a much tougher area. You'll see in a minute. Pharmacogenomics, so knowing your responses to drug or possible side effects. There's a lot of information out there about genetic risk for certain side effects or for dosing. One of the most popular drugs for after you have a heart attack is a drug called warfarin. And if you get the drug wrong, if you get the dose wrong, you could actually be killed by taking too high a dose. So knowing your genotype, knowing the change in your DNA that's responsible for that or is linked to that is actually very valuable. And so I guarantee if you don't learn anything else by getting your genome sequence, you will learn something about your response to certain drugs. And so that is one thing I can say rather assuredly. And the big topic that our lab spends a lot of time on is this one, which is how my story came about, which is how do you bring genomics into managing health of healthy people? And can you do this and what can you learn from this? Because it is a brand new technology. But first I want to run you through some of these cases here. So the one area where genomics is having its biggest impact is in cancer, okay? And I think all of you know this, at least intuitively. Cancer is a genetic disease. In terms of predisposition, there are certain genes, certain changes in genes that leave you very predisposed for cancer. And not only that, once you get cancer, there's a lot of genetic changes that occur that led to you getting the cancer. So virtually every cancer has a genetic basis to it. And they call the non-inherited ones, the ones that get acquired as cells divide along, somatic changes. Now it's thought, this is on average, for a given cancer, whether it's a solid one or a leukemia-type cancer, one in your blood, that there are driver mutations that do this. They're guaranteed driver mutations. And that number is arguable, but it's thought to be on the order of 10 to 20 different changes in your DNA that will drive a cell to grow uncontrollably, i.e. making cancer. Some it's probably less, and others it's a lot more. Okay? And what's been discovered is that every cancer is unique. This is why it's been a tough nut to crack. No one cancer is the same, and it doesn't matter whether it comes from the same origin or not. So for example, if you look at 10 different ovarian cancers, none of them will be the same. And in fact, it turns out it's better to classify cancer based on the genetic changes that have occurred rather than where it comes from. Okay, that is to say, if you have colon cancer, yes, there's a propensity for certain kinds of genetic changes, but more likely than not, by sequencing the DNA, you'll actually see what changes have occurred, and you may be more similar if you have colon cancer. Those changes may be more similar to an ovarian cancer or a lung cancer, something like that, rather than somebody else's colon cancer. And this is why genomics is having so much power, is that what people are doing now is they'll take a cancer patient and sequence the tumor material, the cancer material, and the normal material, and look for those genetic changes that have occurred. And it's challenging because there are many, many changes in there, some of which are these so-called drivers here, but many of them are actually passengers, other changes that have come along the way that aren't necessarily drivers. But nonetheless, by looking at all these changes, the goal is to try and find those that will actually be targetable with a drug. And we've been doing a fair amount of this at cancer, all as part of sort of clinical research. And I'll just show you a fairly recent example that we did with Hongli Ji. And here what we did was we sequenced this individual, and they were amplified, meaning they had increased DNA, a number of loci, but 2 of those loci are shown here. This is a metastatic colon cancer patient, and they had 2 regions that were amplified on this chromosome, a few other regions as well. One of which is in this region called EGFR, and another one is in this one called CDK6, which is the cell cycle gene. So these may not mean much to you, but this turns out to be incredibly interesting because this gene has been mutated in lung cancer and many other cancers. It's rarely mutated in colon cancer, but in this patient it was. 2% of the time it's mutated in colon cancer. But we discovered by sequencing this that this individual did have this mutation. Why is that important? Well, there are actually 2 drugs to this gene. This gene is clearly getting expressed too high a level. There are too many copies of it. And the drug actually inhibits the gene activity. So basically, by sequencing this genome, it suggests that we should be treating this person with a drug against that. We would not have known that had we not sequenced that, and this individual was in fact on one of these approved drugs. It wasn't approved for colon cancer because it wasn't known. It was approved for something else. And that's a story that's recurring over and over again in the cancer field. So sequencing DNA suggests treatments. And I can tell you in virtually every case we've sequenced, we've always gotten some information, if nothing else, about what drug not to use or not so obvious to use because it doesn't make sense. And this is one of the biggest errors of personalized medicine. It's to match drugs for the specific disease, not because it came from an ovary or a colon or a lung, but to match it because they have certain changes that you should be targeting. And this is why cancer is going to be so impactful. Now, I want to tell you these drugs tend to only manage it. In some cases, it's pretty amazing. Patients will, a lot of this is going on at Washington University where they're ahead in certain kinds of cancers, leukemias. And basically, patients have gone into remission based on genomic information, although invariably, like most cancers, it comes back because there are multiple changes that are driving this, and the cancers tend to be heterogeneous. So I think what you're going to see in the future is multiple drugs being used to target cancer. Right now, one can be effective, but in the future, we're going to need multiple drugs, just like AIDS, right? You need multiple drugs to keep AIDS under control. I think that's the way cancer is going to see in the future. We're not there yet, though. Right now, we're at the one drug phase if you find one of these changes. Okay? And so again, the message I want to leave you with is cancer, every individual is unique. And the goal is to find those unique genetic changes, which might suggest drugs, and then use drugs that will specifically target those. And there are all kinds of drugs that are out there now. There's thought to be about a dozen major pathways that are there, but there are hundreds of anti-cancer drugs that people can use. And so that's the approach people are using. Okay, so that's cancer genome sequencing. Another area that's genomics is having big impact is this area of mystery diseases or undiagnosed disease. And they often call them rare diseases because they are rare. A child will be born who have a disease that is hard to figure out. Believe it, it's not that infrequent. It's actually half the time when a severe disease shows up in a child. Half the time you can figure it out because you see something wrong with it. You can guess the test. You can see what's wrong with see where the change is. But the other half the time, you can't. Okay? And these parents who are in these situations are often turning to genome sequencing to see if they can find the root of the problem. Now, it's not always necessarily a genetic change that's causing this, but it could be. And if you see two affected individuals in the same family, meaning, say, two cousins or two siblings that each have the same disease, there's a good chance that it is a genetic disease. Okay? Or if a parent and a child have it, there's a good chance it's genetic. And so again, if people turn to the obvious tests, can't figure this out, what's happening is typically they'll turn to genome sequencing. This is mostly clinical research, although some of this is being done standardly in the clinic. And most of the time, I'll be straight up front with you, we're taking on a number of these cases. We've done at least 30 families or so, more than that if you count related diseases. But most of the time for us, it fails. You sequence their DNA, you can't figure it out. But sometimes it does work, and I'll show you one example for us in a minute. I'll show you one of the early successes. There's been a few spectacular successes where it's led to drug prescriptions that have saved the lives of the kids. This is just one example from Baylor. This is the work of Richard Gibbs and Jim Lupsky. This is a case where there were two twins that were born, non-identical twins. They were born and they were born as so-called floppy babies. And they had a lot of sickness, and they were in and out of the hospital quite regularly. And it was actually the mom who figured out, she read some article in the literature, that they had some disease characteristics that were very similar to something called dystonia. And people treat dystonia with dopamine, and when these kids got to be 5 years old, that's when she figured it out. So it was the mom who figured it out. There's a lesson, by the way, behind all this. And basically, because they figured this out, so they treated the kids with this, it's called L-dopamine, which is used for dystonia. And in 2 days, the kids basically cleared up. Not 100%, but to a large degree. And basically, their health was just dramatically transformed. And so later, when they became teenagers, they weren't 100% cured. There were still some problems there, and they were still getting sick at higher than you would expect frequency. And this is when the genome sequencing technology came along, and basically they sequenced the genome of these two kids, and I believe they did the parents as well. But the net result was they found a change, inactivating mutations in this gene here. Okay, now I'm showing you a biochemical pathway. What's relevant about this is a chemical pathway in our bodies that leads to the synthesis of dopamine, which is shown there. So when this gene is inactivated, people don't make dopamine. Okay? So in fact, that said, well, they're probably on the right track. This is knocked out. This gene isn't working right. And that's why giving them dopamine solved their problems, because you got around the defect. Does that make sense? Okay? So but the other thing you'll notice is that's not the only thing this gene does. It makes dopamine, but it also makes some stuff over here, one of which you've probably heard about, serotonin, which is a neurotransmitter, same as dopamine turns out to be. Well, it turns out there is people are used to working with serotonin. They don't usually give people serotonin. They give them the precursor to serotonin. It's this reagent here. Can't quite read it there. I think it's 5-HT if I remember right. Okay. What's that? 5-HTP. Okay. So they actually gave the kids dopamine plus this precursor, and that actually cured all the symptoms. So basically, these kids, as far as we can tell, they're perfectly healthy, and that's because part of it's because they actually figured out this mutation, and then they deduced what else would help improve the case of the kid. And there's another spectacular case where there's a child who was almost certainly going to die. He's in and out of the hospital, and they sequenced his genome. It's actually the first case, and they discovered he had a mutation in his immune system. So they did an immune system transplant and basically cured the kid, and who's alive and healthy and quite happy now. Although yeah. What's it? These kids are on medicine. Yeah, their health is managed. That's correct. Okay. Although I would argue probably for 3/4 of the people in this room, your health is managed too. Okay. Okay. I may be wrong about it. I won't ask for a show of hands there. All right. So anyway, but I would argue that's generally true for disease anyway. That is, you're managing it. You're never truly curing it. I mean, one could argue the way to cure it is to go in and put a good copy of the gene in these kids, and we're nowhere near as close to that now. All right, but maybe in the future for certain diseases that's important. Okay, here's a case that we've done recently where there was a child. Again, we were trying to figure this one out, and this will illustrate the problem, where we sequenced the DNA of the child and the mother and the father. And again, this is a very typical case. You just get a sea of changes that you detect in their DNA, and then you're trying to figure out which one's responsible for the disease. This is a very typical case for us. We found, based on the child had these severe developmental delays, and we found a whole list of genes that could be responsible for this. We initially zoomed in on one gene, but we couldn't find any biochemical changes that suggest that that was in fact the one. The net result is that in the end, we only figured out the correct gene because another child came along, someone else sequenced, and they had a change in a different, we thought it was this gene. Turns out it was a different gene on this list. And when someone else sequenced the second child that was very much like this first one, they had a change. It was in the second gene. And by making that match, we said, aha, that must be the causative gene. And so this is where this field is going. We just don't have enough information most of the time. You really need to see the thing twice in two different people in order to make this clear what's happening. Now, if you have a very large family, you can figure it out. If you take distant cousins and things, you can actually sequence their DNA, see what they have in common, and then you can sort it out. But in the absence of that, it can be very, very hard unless it's a very specific disease with a very specific defect like the last one I showed on the previous slide. So there are a lot of challenges. And again, so I don't want to mislead you. This works some of the time. I would say in the 5 cases where we figured out what the causative gene is, it hasn't led to any therapeutic treatments, although it has actually been very satisfying to the parents to know what the underlying cause of the disease is, especially for family planning purposes in some cases. So this is, again, where the field is. Most of the time we don't figure it out. Most of the time we get a short list like this of possible candidates, and then we need follow-ups. Okay. So that's kind of where genomics is hitting impacts on health for a disease. But one area, the area that interested me the most, we do a lot of mystery diseases, and we like to try and take on these challenging cases and see what we can learn and see if we can help people with this kind of information. But the way I see it, the future is really not getting your genome sequence after you get the disease. The goal is to get your genome sequence before you get the disease, see what's predictive, catch it early, or even try and manage your health, you manage what you do so that you never get the disease in the first place. That would be the best scenario.